right, thank you. If you've ever visited one of Charleston's salt marshes, you've probably noticed this unassuming green-brown slime that seems to be growing on the sediment. Well, this green-brown slime is most likely benthic microalgae. And benthic microalgae, as the name implies, is microscopic algae that grows on the sediment or the benthos. And it turns out it's actually really important for our salt marsh ecosystems because it helps cycle nutrients, it helps provide all sorts of things. It stabilizes the sediments, and it's a major food source for a lot of the organisms that live in these salt marsh environments. And since it's so important, I decided to study how it changes or how communities of these benthic microalgae change from one time period to the next. And to do this, I had to ask several questions, the two main ones being, what type of benthic microalgae are present at these different time periods? And also, how much of this benthic microalgae is present? So of course, to answer these questions, I had to have some benthic microalgae. So I went out into a Charleston salt marsh and used a modified syringe as a sediment core to collect sediment samples 3 hours before low tide, during low tide, and then 3 hours after low tide. And I did this on both spring and neap tide, so about 8 to 10 days apart, in order to account for different lunar phases. And this was all done on July, October, January, and April in order to account for the different seasons of the year. Now, each of these samples were collected in basically a duplicate. So one of those samples was used to extract DNA, which we then processed and then sent to be sequenced. And this is how we're going to identify what type of benthic microalgae are present. The second sample was used to extract a pigment called chlorophyll A. And this is a photosynthetic pigment that we can find in our benthic microalgae that is used, or it can be used when measured as sort of a proxy or a rough estimate of how much benthic microalgae are present, or their biomass, if you will. So we're still waiting for the DNA aspect, but we do have those chlorophyll A measurements. And it turns out we see significant differences over season. And we're hoping that we can pair the chlorophyll A data with that DNA data that we collected. And we're hoping that that will give us an in-depth look at how we change over, or how those benthic microalgae change over time periods, which we can then apply to larger scale salt marsh ecosystems. And then that will allow us to understand how to protect those in the future. Thank you.